In this video, we are going to talk about the first step in the central dogma of molecular biology, that is DNA replication. And it is one of the most important mechanisms which is going to start the protein synthesis in our body. DNA replication is the process in which the double-stranded DNA structure makes multiple copies of itself. It is a biological polymerization which proceeds in the sequence of initiation, elongation and termination, and it is catalyzed by enzyme. The DNA polymerase is the main enzyme in this replication process along with other such enzymes. First, we will see what is this DNA polymerase. The enzyme here involved in the replication is called as DNA dependent DNA polymerase. The main purpose of this enzyme is to catalyze and regularize the whole process of DNA replication with the support of other enzymes. Deoxyribonucleoside triphosphates are the substrate as well as the energy provider for the replication process. DNA polymerase is of three types: DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Each take care of some special roles in the replication process. For example, DNA polymerase 1, it is a repairing enzyme which repairs the DNA. It especially does it by three activities. First is 5' to 3' polymerase activity. Next one is 5' to 3' exonuclease activity. Next is 3' to 5' exonuclease activity. The second enzyme, that is DNA polymerase 2, is responsible for the extension of primer and proofreading. We will see what this is proofreading in the upcoming slides. The third enzyme, that is DNA polymerase 3, is responsible for in vivo DNA replication. So basically, DNA polymerase is the most important enzyme in the replication process. Now we will see what are all the other enzymes that involved along with this DNA polymerase. As I already mentioned, DNA polymerase, the main role of this DNA polymerase is it matches the correct nucleotides and joins the adjacent nucleotides to each other. And basically this DNA polymerase, we call it as a builder guy, because like how a builder builds our house using bricks, similarly DNA polymerase builds the new strand using the template strand with the help of nucleotides, like how the builder uses a bricks. But one small difference is here the DNA polymerase uses only 4 specific nucleotides, that is A, T, G and C. And second thing is it has to match the exact complementary pairing. Already we have studied what is complementary pairing in the unit 2. So complementary pairing means for A, T is the complement nucleotide and for G, C is the complement nucleotide and vice versa. So this is how DNA polymerase acts. And there is one more special thing about this DNA polymerase, that is it builds the new strand only in 5' to 3' direction. The second enzyme is helicase. What is this helicase? Helicase is the most important enzyme in initiating this replication process because it does the unwinding of the DNA to start the replication process. We know DNA is a double-stranded structure and it is like supercoiled in nature. So this double strand structure, which is like winded very well, has to be unwinded to start the replication process, and it is done by this enzyme helicase. To support the helicase, there is another enzyme which we call it as gyrase or topoisomerase. This topoisomerase relieves the torsional strain in the DNA molecule. So together this topoisomerase and helicase, they help in the unwinding of DNA so that DNA polymerase can start its function. The next enzyme is primase. Primase is providing the primer to the polymerization purpose. So basically you need a starting molecule for producing your new strand, and this starting molecule is provided by this primase, and the starting molecule we call it as primer. So primer are basically short length sequence of nucleotides which is going to act as the base molecules for the new strand. The next enzyme is SSB proteins. Basically, it's not an enzyme, it is a set of proteins which helps in the replication process. SSB refers to single strand binding proteins. So why this is very important? For example, we know that helicase and topoisomerase, they are going to unwind the DNA strand. So what if the unwinded DNA strand again winded back? So to avoid it, you are going to use this SSB proteins. So these SSB proteins are basically to keep the DNA single stranded after it has been melted by the helicase. So it avoids the DNA single strand that was separated by helicase to rejoin back. The next enzyme is ligase. What is the role of this ligase? Basically, when this new strand is formed, there will be few nicks and few gaps available. These gaps and nicks has to be fixed properly to obtain a complete strand, and this is done by ligase. So basically we call ligase as a gluer guy because this ligase joins the gaps and nicks. Next one is exonuclease enzymes, and this exonuclease is mostly done by the DNA polymerase 1 because already we have seen in the previous slide that DNA polymerase 1 has exonuclease activity. These are nothing but a group of enzymes that remove the nucleotide base from the end of a DNA chain, which is very important for this replication process. The last one is telomerase enzyme. This is an enzyme found especially in eukaryotic cells which adds a specific sequence of DNA to the telomeres of the chromosomes after they divide, which helps in stabilizing the chromosomes over time. So lots of enzymes and proteins together, they help out in this replication process. Now we move to the 3 stages of replication. The 3 stages are initiation, elongation and termination. The first stage is called as initiation because in this stage the replication process is initiated. The first step in the DNA replication is relaxation of the double helical structure of the DNA, and this is done by the DNA topoisomerase, or we call it as gyrase. This enzyme will cause the DNA molecule to unwind to a point at which another enzyme called helicase will begin to separate the 2 DNA strands. So both these topoisomerase and helicase works together in separation of the double-stranded DNA. So you get a 2 single-stranded structure, and these 2 single-stranded structure acts as the template for the formation of the new strand. This structure where the double-stranded DNA gets into 2 single-stranded structure, we call it as the replication fork. This replication fork is the site at which the DNA replication actually starts. Since the replication is like bidirectional, that is, it proceeds in both directions from the origin, there are actually 2 replication forks for each replication origin. As the helicase enzyme moves over the double-stranded DNA and opens it up, a protein called single-stranded binding protein, or in short SSB proteins, is bound to the newly single-stranded DNA to stabilize the integrity of the replication fork. Already we have seen why we need this SSB protein, because the single-stranded structure which has unwinded should not rewind and form the double-strand structure again, which is established and secured by this SSB protein. So this is the structure of the replication bubble or the replication forks that are formed during the replication process. So this is the first stage which initiates the replication process. Now the second stage, we call it as elongation. So elongation is the process where the new strand of the DNA is formed using the template strand that is separated in the first step. So basically here in this step, the parental strands will act as the template for newly synthesizing daughter strands. It is to be noted that elongation is unidirectional. So when I say unidirectional, we already know DNA is always polymerized only in 5' to 3' direction, because it's one of the important condition of the DNA polymerase. So only in the one strand the DNA is going to be formed, and this particular strand we call it as the leading strand. But the problem is the opposite strand is going to happen in a reverse direction due to which we call it as a lagging strand. In this particular lagging strand or the discontinuous strands, due to the lagging or the reverse effect, there is going to be occurrence of fragments called Okazaki fragments, and these Okazaki fragments later will be joined together back by the DNA ligase. If you see this image clearly, already as I said in the initiation step, topoisomerase along with the helicase enzyme, they unwind the double-stranded DNA structure. So you get two single-stranded DNA which acts as the template strand. So now you have your RNA primer which is added by the DNA primase enzyme. Once this primer is added, from which the DNA polymerase enzyme is going to start the formation of the new strands, and this formation is made by adding the complementary nucleotides for the template strand. So in this two strands, one strand we call it as the leading strand. So this leading strand is emphasized because it moves from 5' to 3' direction, which is the actual direction DNA polymerase expects to go on. So because of which there won't be any intermediate fragments formed, and it will be moving in the direction towards the topoisomerase or helicase. But in the opposite strand, the formation will happen in the reverse direction, that is opposite to the direction of topoisomerase and helicase movement. So because it's happening in the opposite direction, the fragments that are formed will be having gaps in between. So whenever there is a gap, the next level should be starting with again an addition of RNA primer, due to which lots of fragmented segments are formed, which we call it as Okazaki fragments. So this fragment that is formed, we call it as the lagging strand. So basically in the elongation process, we get two newly synthesized strands, in which one we call it as leading strand and the another one we call it as the lagging strand. The last step is termination. So in this particular termination process, after the synthesis, the exonucleases will remove all the RNA primers from their original strands. Once the primers are removed, the DNA polymerase will check, or we call it as the proofreading activity, where it will check whether it has added the right nucleotides which is complementary to the template strand, and it will check for any errors. Once the proofreading is over, the DNA ligase enzyme will join the Okazaki fragments to form a complete single strand in the lagging strand. Once all these things are performed, the ends of the parent strand consist of a repetition of DNA sequence known as telomeres, which acts as the protective caps at the end of chromosomes, preventing the fusion of the nearby chromosomes. And these telomeres are synthesized by a special type of DNA polymerase enzyme known as telomerase. This telomerase catalyzes the telomere sequence at the end of the DNA. Once completed, the parent and the complementary strand coil into a double helical shape, producing two DNA molecules, each passing from one strand from the parent molecule and one from the new strand. So this is the final step where the leading and lagging strands have been fixed by the DNA polymerase and the proofreading was over, and finally you get two separate DNA strands. This process is the first step in the central dogma of molecular biology, where the one molecule of the DNA gets converted into two molecules of DNA.